Okay, you can see that?
Yeah, yeah, okay. Can we have the first slide? So for
Yep.
this theme, the proposed topic is diversity and functional gene analysis of gut microbiome in Nigerian patients with Parkinson's disease. It's a new study because it's a proposed topic. And on it, we have I'm Dr. Nwaokorie, Francisca, myself, Ifeoma, and Lillian. So we are the four on the proposed topic. Next slide, please. Next slide, yes. So this is for the narratives. We are going to provide a baseline data on gut microbiome of patients with Parkinson's disease for the first time in Nigeria. There are studies on Parkinson's disease, but very few studies, and we do not have any study on microbiome of patients with Parkinson's disease. So this will provide a baseline data for it. It will also provide a reference data, and then whatever we get out of it will aid in the management of the disease. So, and it will also help in predicting and monitoring the disease. As a result, in the future, we'll maintain a cohort of humans for future research. Can we have the next slide? Next slide. So Parkinson's disease is a long-term degenerative disorder of the central nervous system that mainly affects the motor system. And it actually affects the elderly most times, above 65 years. And when it comes earlier than that, it is referred to as an early onset. In the world, we have about 1.55% of world population having the disorder. And then we have about 4.70 to 23% dying from the death. In sub-Saharan Africa, the prevalent rate is about 10 to 235 in 100,000. In Nigeria, we really have few data. And one thing about Nigeria is that Nigeria is the most populous country in the whole of Africa. And we have about 5.25 million people who are aged above 65 years. So we have few data on Parkinson's disease, and many of them, some of them are affected by Parkinson's disease. Unfortunately, they are not easily or early diagnosed. So some of them go unnoticed. But now, when some of them come to the hospital, you find out that they end up having Parkinson's disease. So a high number of these adults have Parkinson's disease, and we do not have any data. So it's just a few cases or few reports that we have. And it's been found out that, go to the next slide, please. Next slide. Yeah. Parkinson's disease, because it is a motor neuron disease or that affects the nervous system, we find out that many of them have these freezing gaits when it affects them. Some of the symptoms that we have, some of them have speech problems. They have difficulty in swallowing. Then some of them have reduced ability to smell. Then we have personality changes. Then some have paresthesias, that is their hand and their palms. They have tingling palms that pains them. Some result into depression. Some have orthostatic hypotension, where they get up on the bed, they have hypotension, their pressure drops. It ends up in hallucination, and so many of these symptoms in these elderly people. Next slide, please. Next slide. So Parkinson's disease is associated with formation of Lewy bodies in the central nervous system. These bodies may spread from the gut to the brain, via the enteric nervous system to the vagus nerve, and then the CNS again is affecting the motor system. And that is why you have so many of those symptoms. But because of the communication between the gut and the brain, it's been found out that there is a correlation between the gut microbiome of patients with Parkinson's disease and the gut. So this particular project, we delved into many studies and we found out there are studies, but none of them has really dealt on it. And despite that, there is bacterial diversity that has been found in patients with Parkinson's disease, but we do not really have anything about it. So as a result, we decided that we'll probably go into this and find out whether there is even a difference between patients with Parkinson in Nigeria, because the kind of food or the nutrition in Nigeria is slightly different. So we'll be able to check the metabolic functions and see what is going on there. Is their gut affecting their own? Is it the same thing with what we found in other parts of the world? So we want to check whether the microbiome signature of Parkinson's disease might allow the development of biomarkers that can trace or they can monitor the development of Parkinson's disease. Next slide, please. Next slide. So our research question will address what is the spectrum of microorganisms that are found in the gut of patients with Parkinson's disease. What differences occur in the microbiomes as a result of diet and medications for management of Parkinson's disease? What are the most commonly occurring gut species in different population groups in Nigeria, and factors mitigating alterations? So from our research questions, we'll be able to, when we have our aims and then our objectives. That's what we want to go by. The methodology will be handled by Francisca. Hello, can you hear me?
Yeah, I'm here. Tayo, I can pick up from you. Okay. Yeah, based on what Tayo has done to give us a background on Parkinson's disease and as it relates to the gut, our area as medical microbiologists is to look at these microorganisms and what do they do and what are they producing that is actually affecting our central nervous system. So majority of them are the metabolites that they produce, which affects the neurons. So in the study we're proposing to do, we're going to make it like a cross-sectional study for a start, and then we can expand on trying to monitor the progression. So our study population will be aged people that we're going to match age by sex with control, so that we'll compare those who don't have Parkinson with those who have Parkinson's disease and be able to correlate which microorganisms are present. Then we'll look at the food that they eat, which we can capture using an environmental or clinical questionnaire to be able to relate with these clinical symptoms and the environment and the microorganism that we are finding from, we would likely get from our study. So our sample size is going to be pragmatic. That means anybody presenting within that period of time of the study, we are proposing for an initial one of about 2 years and then 5 years. And our protocol is going to be standard protocol as designed by the Human Microbiome Project, which is open. And we've been able to assess it to know inclusion criteria and exclusion criteria to follow. We'll be able to do DNA extractions using a microbiome extraction kit and a fecal DNA extraction kit. Luckily, we've tried these kits and they've worked for the work we have done so far. And we're gonna do sequencing using a PacBio. PacBio look at fragmentation of DNA to be able to give us their whole genome. Then thereafter, we can use it to see the diversity using a bioinformatic analysis. Then we'll look at descriptive statistics and then compare with the clinical findings and relate that and correlate it with what we have. We should be able to also go to gene banks by the gene bank, and then we can be able to look for the one in Europe, look at the one at US, then compare with what we have as our unique microbiome in Nigeria to see, is there any difference in what we are getting? Does it have anything to do with the food we eat and then the people around here? Next slide, please. Next slide. Okay, so what is the expected outcome? By the time we start this study, of course, we have formed a team of our members who are going to work with this work. We are also going to network with experts in the field who are able to get the patients, examine them, and make their contribution. Then we'll look at the relative abundance, see if we can be able to identify any species, and then look at the functional genes that we can also get. And those functional genes can be used as biomarkers that can be used to actually identify those with Parkinson's disease as it relates to Nigeria. So this information should be able to provide a data tool for disease database which we can use as big data to be able to predict and then model the Parkinson's disease in Nigeria. At the cost of also doing the work, which we propose to do, we should be able to train, organize workshops, seminars, symposium, just like we are doing now, to be able to disseminate the information for those who will learn from it. Then we should be able to have peer-reviewed publications. And if we're able to get grants that can also support students for master's and PhD, we should be able to roll out thesis and dissertation from that too as well. Next slide, please. So what is our plan? We flow out the proposal, and the area we want to look at is open for input. We have a short-term plan of looking at the gut microbiome and Parkinson, colorectal cancer. That's the long-term plan. We also need to also extend. Then we have the short-term plan that can lead to a long-term plan. And finally, we might be able to look at the way it relates to women, the vaginal microbiome, and relate it with what we have seen with the gut and the kind of diseases that might actually be a problem to women. Next slide, please. So this is one of the publications that I've just done so far with our team. We looked at the baseline. What is the gut microbiome of an average healthy Nigerian, to be able to establish what is the basis. So if we now look at disease conditions like Parkinson's disease, we should be able to compare. Did we have something different, or they are also the normal organism that we found in Nigeria? This work is actually a mentored research, which is a step down from a project that is being sponsored in Nigeria by NIH. Next slide, please. So what strategy are we also having to do this? We should be able to see collaboration, and we hope that this presentation will open up to GWIS members internationally who want to be part of it, or even a mentor that can help out with the study. We should be able to see grant nationally and internationally. We should be able to design a project that will accommodate mentorship and exchange programs between our members, which can help us to build our career progressively and promote whatever vision we have in the field of science. As long as GWIS is concerned, that is the aim, to be able to let international women be involved in research and be able to project what they know how to do in terms of global recognition and impact. Next slide, please. Thank you so much. That's all we have for now. And thank you for the audience.
No, thank you very much.